Welcome to Science with Tal. In today's video, we will continue our conversation on the fundamental mechanisms of neurons. In the last video about signal propagation, we have covered important concepts such as the electrochemical gradient, ion channels and transporters, passive membrane properties, and the Hodgkin-Huxley model of the action potential, to name a few. We've also covered a lot of equations that describe the equilibrium potential and the membrane potential, the passive membrane properties and the associated time and space constants, as well as equations describing the probabilistic nature of voltage-gated channels. I've also introduced some important experimental techniques, such as the voltage and patch clamp. In this video about signal transmission, it will often happen that we will have to come back to some of these concepts, but I will make sure that you know the key elements that have to be known when we encounter topics that require specific background knowledge that we've covered previously. With this being said, let's go back to where we left off. Alright, now that we understand how the signal travels through the neuron, we now need to understand what happens when that signal reaches the axon terminal. I mentioned in the very first overview that neurons communicate at synapses, which are the sites of signal transmission. I've also mentioned that the cell sending the signal is referred to as the presynaptic cell, and the cell receiving the signal is the postsynaptic cell. Also, recall that the space between the two neurons is called the synaptic cleft. There are two types of synapses that can be found in the body: chemical and electrical synapses. All the synapses that I've shown for the moment were chemical. To see how these two types of synapses differ from one another, let's first go over electrical synapses. Electrical synapses are found between neurons and between glial cells, and are characterized by the fact that there is a cytoplasmic continuity between the pre- and postsynaptic cells. This continuity is facilitated by gap junction channels. One gap junction channel is made out of two connexons that are each embedded in the membrane of the pre- and postsynaptic cell. One connexon is made out of 6 connexins, hence one gap junction is made out of 2 connexons or 12 connexins. Connexins are a very diverse group of transmembrane proteins. In the human genome, there are about 21 different types of connexin genes, which allows great diversity in the gap junction channels. The 2 connexons together form one large non-specific pore that allows current to directly flow through. The big diameter pore also allows metabolic signals such as CAMP, small peptides, as well as inorganic anions and cations like calcium to flow through. Because the cytoplasm is continuous through these gap junctions, signaling in electrical synapses can be bidirectional, but due to the diversity of connexins, some gap junctions only allow to flow unidirectionally. Gap junction channels aren't always open, however. Most gap junction channels can close in response to a low pH, elevated calcium, phosphorylation, or voltage. But that depends obviously on the type of gap junction. Another consequence of the continuous cytoplasm is the fact that there is essentially no delay in terms of the response from the postsynaptic cell after a signal is sent from the presynaptic cell. For example, if an action potential travels through the presynaptic cell, and after passing through the gap junction the current from the action potential is above the threshold for the postsynaptic cell, then an action potential can be fired from the postsynaptic cell almost instantaneously. A good example of this synchronized firing occurs in the cardiac muscles, which are cells that make up the muscles of the heart. In this very crude diagram of the cardiac muscles, you can see the cardiac muscle cells, which are connected by these purple bands called intercalated discs. These discs contain gap junction channels that allow the flow of current, metabolites, and etc. to flow through. When action potentials travel through these cells, the small delay in rapid transmission of the signal is propagated in the cardiac muscles with synchronicity and produces rhythmic contractions of the heart. In neurons and glial cells, electrical synapses have functions that are less understood, but regardless, you can see that the very rapid transmission that gap channels provide can be very beneficial depending on the need of the body. Now, when it comes to chemical synapses, there is no continuity in cytoplasm between the two cells, and the synaptic cleft is about 5 to 10 times larger than electrical synapses. To establish some useful terminology on chemical synapses that we will use for the rest of the video, let's go over the general process of how chemical synapses work. Everything begins when the action potential reaches the axon terminal, where neurotransmitters are synthesized and packaged into synaptic vesicles. This region in the presynaptic cell, where neurotransmitters reside, and are ready to be released is usually referred to as the active zone. The depolarization carried by the action potential allows to open voltage-gated calcium channels. Because of the steep calcium concentration gradient, when the channels open, calcium rapidly enters the active zone. The entry of calcium causes the fusion of the vesicles with the membrane and the subsequent release of neurotransmitters in the synaptic cleft. This release of neurotransmitters is also referred to as exocytosis. As neurotransmitters diffuse in the cleft, they bind to receptors on the postsynaptic cell, which are located in a region called the postsynaptic density. There are two main types of receptors on which neurotransmitters can bind: ionotropic and metabotropic. Ionotropic receptors are essentially the ligand-gated ion channels that I have introduced when I talked about the general properties of ion channels. When these channels open, the ions that the channel is selective to can flow in or out depending on their driving force. As ions enter or leave the cell through these channels, the membrane potential of the postsynaptic cell is either depolarized or hyperpolarized. For that reason, these channels provide a rapid indirect path for ionic efflux and influx. The impacts of ionotropic receptors usually last for a brief period, which is usually the time that the membrane readapts to its resting potential. Metabotropic receptors, on the other hand, have more indirect effects in comparison to ionotropic receptors. When they are stimulated, they produce second messengers such as cAMP or diacylglycerol that usually activate protein kinases that have a multitude of different physiological impacts. One such impact is the phosphorylation of other ion channels, which regulates their opening or closing. Because of these indirect effects, metabotropic channels provide amplification and modulation mechanisms through second messengers that can impact the cell for considerably longer periods than ionotropic receptors. Anyhow, this is just a brief introduction. We will get into the fine details later when we will discuss their effects more concretely. You will notice that in this general model of the chemical synapse, the transmission is unidirectional, but we will see later some examples where this blurs out a bit. For the most part, however, you can consider that the signal is unidirectional. Moreover, due to all these steps in signal transmission, chemical synapses have a delay of about 1 millisecond that is very notable in comparison to electrical synapses. Based on what we covered, when we compare the two synapses, you will see that the two are very different and have their respective nuances. As far as this video goes, we will not extensively go back to electrical synapses because they are not as utilized in the body as chemical synapses. And to be quite honest, in this introduction I've covered the bulk of what we know about them. So with this being said, let's get better insight on how our knowledge on chemical synapses came about by considering the synapse between the motor neuron coming out of the spinal cord and the skeletal muscle that it innervates. When we take a deeper dive into what the skeletal muscle is, you will see that the muscle is made out of many muscle fibers, and those are what the motor neuron innervates. This synapse between the muscle fibers and the motor neuron is called the neuromuscular junction. The neuromuscular junction obviously has very important impacts on the contraction of muscles, but as I said, in this video we want to understand the basis of where our understanding for chemical synapses came from, and for that reason, we won't discuss muscle contractions in this video. This topic will be covered in a video about the motor system. We will only consider the neuromuscular junction as this simple system of a motor neuron transmitting a signal to the muscle fiber to model chemical synaptic transmission. Now, one might ask, what is so compelling about the neuromuscular junction that we want to base the chemical synapse model on it? The first advantage of this synapse is its size. In comparison to synapses in the brain, which are measured on the nanometer scale, the synapses at the neuromuscular junction are on the micrometer scale, which makes them about 1,000 times larger. The second advantage is its simplicity relative to synapses in the brain. Indeed, the neuromuscular junction operates on a simple system of one neurotransmitter and one ionotropic receptor, whereas systems in the brain can become very complicated as many neurotransmitters and many different types of receptors can get involved. The third advantage of this synapse is its accessibility, and this should be obvious from the first point given that they are considerably bigger and usually more isolated. These three factors together make the neuromuscular junction a very suitable site for analysis and allow electrophysiological recordings to be made without damaging the structure. Now, the way I will go about this section will be a bit different than how it is typically covered. Instead of first going through the experiments that gave insight into the process and then constructing the model, I will first go over how chemical transmission at the neuromuscular junction is currently understood, and then we will look at the experiments with a sort of introspective lens to understand how they've managed to build what we know today. So, for terminology's sake, the many branchings leaving the axon form what are called synaptic boutons, which essentially correspond to the sites of chemical transmission. The synaptic boutons are enwrapped in Schwann cells, the same type of cells that cover the axon. The summed region covered by these boutons is usually referred to as the end plate. Now, let's consider one side of transmission which looks a bit like this. Alright, first, let's discuss how the neurotransmitter is packaged in the vesicles by considering this small patch of the terminal. The neurotransmitter inside these vesicles is acetylcholine. This neurotransmitter is synthesized in a rather straightforward reaction. The enzyme choline acetyltransferase catalyzes the formation of acetylcholine from choline and acetyl-CoA. Acetyl-CoA is made from the metabolic oxidation of glucose by the mitochondrion, and choline enters the cell by the choline symporter that carries sodium inside as well. To get a transmitter inside the vesicles have a vesicular acetylcholine antiporter that uses the energy from proton export to get acetylcholine inside. In a later section, I will explain in more detail how the proton gradient is generated and what other important proteins the vesicle carries. Note that every vesicle has this transporter, even though it's not shown on the schematic. As you can see, everything from the synthesis to the packaging of the neurotransmitter is directly done at the terminal to ensure efficiency. With this step covered, we can now consider how the transmitter is released. The release of acetylcholine follows exactly the same steps as we've established previously when we discussed the general properties of chemical synapses. The action potential causes the opening of voltage-gated calcium channels, calcium enters, and causes fusion of vesicles. The exocytosis of acetylcholine then causes them to diffuse in a cleft and bind to receptors. The receptor on which acetylcholine binds is called the nicotinic acetylcholine receptor. This receptor is a pentameric ionotropic channel. At the neuromuscular junction, the distribution of subunits is 2 alpha, 1 beta, 1 delta, and 1 gamma. I mentioned this distribution because the nicotinic acetylcholine receptors in the central nervous system have a different distribution, 2 alpha and 3 beta. The channel has 2 binding sites for acetylcholine that are located between the alpha subunits and the neighboring gamma and delta subunits. To open this channel, the 2 binding sites must have acetylcholine inside. When the channel opens, mainly sodium and potassium flow through according to their driving force. Thus, the nicotinic acetylcholine receptor provides a path for both potassium efflux and sodium influx. The pore size of the channel has been found to be considerably larger than the previous selective channels we have discussed, and thus, it explains why sodium and potassium can both flow. Even calcium can flow through this channel, and for that reason, it is usually referred to as a non-selective cation channel. I will later explain what experimental technique allows to discover which ions go through the channel when we have a better understanding of some prior concepts associated to it. On a side note, I want to mention that the reason this channel is named the nicotinic acetylcholine receptor is because nicotine is a substance that is capable of binding to the channel and opening it such that ions can flow. Such compounds that can bind to channels and open them are referred to as agonists. Another example of an agonist is obviously acetylcholine, but usually it isn't referred to as an agonist because it is the main substance that physiologically binds to these channels. We would technically say that nicotine is an agonist to acetylcholine for this receptor. On the other hand, you can also have compounds that bind to the channel and do not open it. Such compounds are called antagonists. 2 common antagonists for this channel are alpha-bungaratoxin and curare. These compounds are often used to reduce the action of acetylcholine receptors in certain experimental conditions, and we will later cover some experiments that use curare. Back to the schematic, now that we have a good idea on how these postsynaptic receptors function, We can now understand that the opening of these nicotinic acetylcholine receptors by the binding of acetylcholine allows sodium to enter and potassium to exit the cell. It turns out that the channel is a bit more conductive to sodium, which causes a net depolarization that passively propagates in the muscle fiber. Something I haven't mentioned yet, which is one of the most notable features of the neuromuscular junction, are the multiple folds it has on the postsynaptic side. The reasoning behind these folds is that they considerably increase the surface area in which the receptor channels can be embedded. Accordingly, the acetylcholine receptors are concentrated near the crests of these invaginations, while voltage-gated sodium channels are concentrated in the troughs. This simple organization allows the neuromuscular junction to have a low activation threshold, such that the depolarization coming from the receptors easily cause the opening of voltage-gated sodium channels and the subsequent firing of an action potential at the muscle fiber. Remember that this action potential in the muscle fiber ultimately leads to muscle contraction. But we know from our personal experience that we do not want our muscles to constantly contract, so there must be a system in place that takes care of terminating the signal and stopping the opening of the receptors. The way the signal is terminated in the neuromuscular junction is with the aid of acetylcholine esterase. Acetylcholine esterase is an enzyme anchored in the basal lamina, which is a meshwork of collagen and other extracellular matrix proteins, here represented by this blue zigzaggy line. The main catalytic function of acetylcholine esterase is to rapidly hydrolyze acetylcholine into acetate and choline. From this, choline can be directly recycled by the presynaptic terminal to make more acetylcholine, but more importantly, the neurotransmitter is degraded and can obviously no longer interact with the receptors. Now that we understand how the synapse at the neuromuscular junction functions, let's see what experiments helped pave the way for this model. One pioneering experiment that helped establish many properties of the neuromuscular junction came from recordings made by Bernard Katz. To perform the recordings, Katz and his colleagues stimulated the axon motor neuron with an electrode and measured the membrane potential of the muscle fiber with another electrode. What this experiment essentially seeks to understand is how the muscle fiber reacts when an action potential is fired in the presynaptic motor neuron. If we do this experiment under physiological conditions and we keep track of the membrane potential as a function of time, the action potential generated in the motor neuron will produce an action potential in the muscle fiber after a small delay. Based on the chemical transmission system that we've just described, this response from the muscle fiber makes sense because we expect muscles to efficiently respond and contract in response to messages coming from motor neurons. This recording, however, hides the actual depolarization from the nicotinic acetylcholine receptors that took place to generate the action potential. The actual depolarization through the receptors looks something like this. As you can see, this red shaded curve, called the excitatory postsynaptic potential, is above the threshold, and that's why it generates an action potential. But this is not something that we can technically see because the recordings would just show the curve for the action potential. Because this event occurs at the end plate potential, the specific excitatory postsynaptic potential is also referred to as an end plate potential, which is the terminology I will use to describe EPSPs at the neuromuscular junction. To isolate the end plate potential, one can apply the drug Curare in the system. Obviously, in real life, the neuron would not stand up straight like this, but you have to imagine that the experimental setup has this Curare influence on the muscle fiber. Recall that Curare is an antagonist and prevents the opening of acetylcholine receptors. Consequently, in this system, when an action potential is sent to the muscle fiber, the end plate potential is reduced below threshold and does not generate any action potentials in the muscle fiber. The end plate potential then decays with time. The scientists behind the experiment now had a direct measure of the receptor's influence on the membrane potential. But going back to the physiological state, a very interesting result from these recordings that Katz noticed was the occurrence of small end plate potentials that happen spontaneously without any presynaptic stimulation. These random depolarizations were accordingly labeled miniature end plate potentials, or simply MEPs. You will notice that these miniature end plate potentials are very similar in shape to the actual end plate potentials we get from stimulating the motor neuron. To manually get these MEPs from motor neuron stimulation, Katz and his colleagues used another technique to reduce the size of the end plate potential to try and match the size of the miniature end plate potentials. Their technique essentially consisted in baiting the postsynaptic muscle fiber in a solution of low calcium and high magnesium concentration. When the motor neuron is fired under these conditions, the magnitude of the EPP is reduced drastically and approaches the size of the MEP. What Katz and his team realized is that the smallest magnitude of the EPP that was not zero was identical in shape to the MEPs. The fact that the smallest possible end plate potential was the size of the miniature end plate potential hinted at the fact that MEPs might represent a quantum. In the words of Katz, the hypothesis behind these results is that EPPs are made out of unitary spontaneous MEPs. To support this quantum hypothesis, the scientists performing these experiments assumed that the phenomenon behind miniature end plate potentials were random, and thus they tried to find a probabilistic relation that would fit their experimental data. When we consider multiple trials with this setup, you will notice that sometimes the end plate potential is larger than the miniature end plate potential, here in trial 3, and that sometimes the end plate potential doesn't happen like in trial 4. Such cases are referred to as failures. But this is just 4 trials. There is no obvious relation to take away from here. In the paper from Boyd and Martin, the two scientists illustrate what happens if for many more trials, about 200 more, you plot how many times you see a given amplitude. To make it more clear, let me remake their histogram. On the histogram, you will first notice in the right corner a distribution for the amplitudes of the miniature end plate potential, and as you can see, the most common amplitude associated to it is 0.4 millivolts. Obviously, because of experimental error, not all the recordings gave 0.4 millivolts, so there is a range of values between 0.2 and 0.5, but nonetheless, this is still a very small gap and thus makes the amplitude of MEPs pretty consistent. For these results, we can thus associate a Gaussian curve with a mean amplitude mu and variance sigma squared. Now, to get acquainted with the bigger histogram, let's see how the 4 trials we have fit in the graph. In the first trial, we have a single EPP that has the exact same magnitude as the MEP. If we assume our MEP has a magnitude of 0.4 millivolts, then our first recording would go in this column. In our second trial, we have an EPP that is about twice the size of the MEP and thus goes in this column. In the third experiment, the EPP is a bit bigger than the MEP, but not twice as big, and thus would go in between 0.4 and 0.8 millivolts. Finally, our fourth trial is a failure and goes in the failure column at 0 millivolts. Even though this experimental data already hints at the fact that the EPP is made out of multiple MEPs, or to be more precise, according to this data, the magnitude of the EPP would be a multiple of 0.4 millivolts, we still need a more precise analysis to better support the hypothesis. This analysis is based on a statistical theory derived by Katz and his team. In this model, the letter n represents the number of quantal units that are released, and p the probability that it is released. The letter m represents the quantal content, and it is given by the average EPP size divided by the size of the MEP. For this statistical model, we will assume that the probability to release a quantal unit is small and constant. Also, we will assume that each release occurs independently. To model the theoretical distribution of expected observations, we can use the mean and the variance of the MEP distribution to separate the histogram in different groups. The first group corresponds to the mean and the variance, the second group corresponds to twice the mean and twice the variance, and the subsequent groups follow this trend. For each group, we can model a theoretical curve using the Poisson distribution, which is often used to model the occurrence of successes in a large number of trials. The Poisson distribution is given by this equation, and to find the theoretical distribution, we can multiply it by n, which represents the total number of observations. X is a number associated with the group and m is the quantal content. For each group, we can get a theoretical curve that illustrates the theoretical amount of observations that should be seen. When we add all the curves together, we get a continuous distribution that closely resembles the experimental data. The sum theoretical distribution also gives an estimation of failures which closely matches the experimental data. To summarize everything that we've covered here, the main takeaway is that this theoretical fitting further supports the idea that miniature end plate potentials are the units of end plate potentials. With the discovery of the quantal release, one important question that we need to uncover before we discuss what the quanta are is what exactly triggers the release of these quanta. The intuition that calcium had a role to play in synaptic transmission became apparent as baiting the neuromuscular junction in a lower calcium concentration made the postsynaptic responses much lower, as this experiment shows. More convincing experiments, again made by Katz, that sought to determine what was the signal for chemical transmission, utilized the giant synapse of the squid. This synapse is very convenient as its size allows the insertion of electrodes into the pre- and postsynaptic structures. 2 electrodes are inserted in the presynaptic cell, one for stimulation and one for voltage recording. A third electrode is inserted in the postsynaptic cell to record the voltage there. The third electrode is very important because it provides a way for the experimenter to know if the signal was transmitted or not. When an action potential is generated in the presynaptic cell, the excitatory postsynaptic potential is usually strong enough to cause an action potential in the postsynaptic cell after a small delay. The same experiment can be performed in the presence of tetrodotoxin or TTX. Remember that TTX is a toxin that blocks voltage-gated sodium channels and thus impacts the action potential of the presynaptic cell. In this setup, what the experimenters have noticed is that in successive action potentials, the amplitude of both the presynaptic action potential and the postsynaptic excitatory postsynaptic potential gradually decrease as the effects of TTX get stronger. This result entails that the size of the presynaptic depolarization influences the postsynaptic response. We know from previous discussions that the action potential is mainly governed by the influx of sodium and the subsequent efflux of potassium through voltage-gated channels. The experimenters at the time knew that aspect as well and wanted to see if sodium and potassium also had something to do with the release of transmitters. To see which ions are responsible for signal transmission, the experimenters first used TTX to block sodium channels, and by injecting current in the presynaptic cell, the experimenters still saw an EPSP in the postsynaptic cell. They thus concluded that sodium is not necessary for signal transmission. They then turned their attention to potassium and added tetraethylammonium to the TTX solution to block voltage-gated potassium channels. Here again, after a current is injected, transmission still occurs. The results here are a bit different as the action potentials are sustained throughout the current pulse. This is explained by the fact that the inactivation of potassium prevents the repolarization phase and thus the cell stays depolarized. Either way, it was clear that sodium and potassium are not the ions responsible for transmission. These results made the experimenters shift their attention to calcium. As I mentioned in the beginning, Katz already knew from his experiments on the neuromuscular junction that lowering the external calcium concentration decreased the size of the end plate potential up to the size of the miniature end plate potential. The team also found out that the voltage-gated calcium channels were much more abundant at the axon terminal. And as a reminder, under physiological conditions, calcium is kept at very small concentrations in the cell relative to the outside, which produces a very strong driving force. The conclusive proof that calcium was the signal for transmitter release came from experiments made by Rodolfo Lines and his colleagues, which utilized the voltage clamp technique. Remember that the purpose of the voltage clamp is to lock the cell at the command voltage and send a compensatory current that maintains the command voltage. The compensatory current is a direct measure of how the cell reacts to the change in membrane potential. Lines and his team voltage clamped the presynaptic neuron and applied the same pharmacological blockers to prevent any sodium or potassium current from flowing, such that it isolates the calcium current. They also placed an electrode in the postsynaptic cell to record the voltage there. In terms of the recordings, we have 3 plots. We first have a plot for the command voltage, one for the current in the presynaptic cell, and finally a plot for the postsynaptic potential. All of them are as a function of time. When the presynaptic cell is clamped above threshold, The recordings show an inward calcium current in the presynaptic cell and a rising postsynaptic potential. At higher command potentials, the responses elicited in the postsynaptic cell were bigger. The results also show that voltage-gated calcium channels do not inactivate like voltage-gated sodium channels and stay open as long as the depolarization persists. When a calcium channel blocker such as cadmium is introduced in the system, the presynaptic current and postsynaptic depolarization are blocked. In summary, calcium is often said to be necessary and sufficient for transmitter release. The necessity comes from the fact that if one adds calcium buffers in the presynaptic terminal that inactivate calcium, then even when the action potential depolarizes the terminal and calcium enters the terminal, the buffers will prevent calcium from causing the release of transmitters and thus preventing signal transmission. The sufficiency comes from the fact that if calcium is injected in the presynaptic cell, it can trigger the release of neurotransmitters even without the input of an action potential. Alright, now that we understand that transmitters are released in quanta and that calcium causes the release, we need to understand what the quantas are. The insight that the quanta measured by Katz were coming from the release of neurotransmitters through vesicles became pretty apparent with studies using electron microscopy. In the mid-1950s, electron micrographs of the presynaptic terminal revealed that presynaptic terminals were filled with what they called vesicular-appearing bodies. With these images, Katz put forward the hypothesis that these small vesicles are the quanta that, upon release, cause the miniature end plate potentials. Subsequent studies have then shown that vesicles indeed contain neurotransmitters such as acetylcholine. To support the hypothesis from Katz, pioneering research using the electron microscopy technology was made by John Heuser and Heuser Reese. In their 1979 paper, the team provided experimental evidence that the quanta were indeed vesicles, and they also showed images of exocytosis at the neuromuscular junction of frogs. To capture the exocytosis process, as you can see on the right, the team performed a technique called freeze-fracturing electron microscopy. In a nutshell, freeze-fracture electron microscopy allows the experimenter to visualize features of the cell membrane by seeing the distribution of proteins in the membrane. To do so, the cell is first frozen and then fractured. Here, the fracture essentially splits the phospholipid bilayer in two halves by using a sharp microtome knife. In terms of freeze-fracture terminology, the lipid layer near the cytoplasmic region, as viewed from the hydrophobic tails, is named the P face. After cleavage, the surface is shadowed with a platinum film, which produces a replica of the surface. The replica is then coated with carbon to increase its stability. After the membranous tissue is dissolved from the replica can be imaged in the transmission electron microscope. The subsequent image captured by the microscope is what you see on the right. To make sure they would be able to visualize the vesicles fusing, the team applied the drug 4-aminopyridine, or 4-AP as abbreviated in their description. This drug is essentially a voltage-gated potassium channel blocker, and as you know, blocking voltage-gated potassium channels will reduce the repolarization and thus increase the calcium influx, which allows them to increase the probability that vesicles will fuse. In Reese and Heuser's actual protocol, there are obviously a lot more steps in between, but the general guidelines of what they did to get these images follow what I just explained. In these electron micrographs, you can directly see the P face of the presynaptic motor nerve. In the first image, the first panel corresponds to a quick frozen control. The second panel corresponds to a freeze image made 5 milliseconds after stimulation. As you can see in the second panel, there are deformations in the membrane immediately after synaptic activity. The team proposed that these were vesicles undergoing exocytosis. The small dots around are thought to be voltage-gated calcium channels. To prove that the electrophysiological quanta that we know as miniature end-plate potentials are indeed caused by the fusion of one vesicle, the team tried correlating the number of observed vesicles fusing with the elicited response at the postsynaptic cell. Indeed, as they were freeze-fracturing the presynaptic terminal, they were also recording the postsynaptic potential in parallel as neurotransmitters were released. In this figure, the y-axis corresponds to the number of vesicles in the act of fusion that they counted, and on the x-axis, the theoretical amount of vesicles that should have been released to produce the response they recorded. As you can see, as the 4-AP concentration increases and more transmitters are released, the relation between the x and y-axis is linear and entails that a vesicle is pretty much equivalent to one quanta. In all, this provides strong evidence that transmitters are indeed released through exocytosis, and the quanta as measured by Katz are vesicles filled with a somewhat constant amount of neurotransmitters. In an earlier paper in 1973, Heuser and Reese were able to show experimentally, again at the neuromuscular junction of frogs, that the vesicles are recycled by endocytosis. An important question for them to answer was, under constant stimulation and presumably fusion of the membrane, how can the presynaptic terminal keep a constant surface area if the vesicles area adds onto it? To demonstrate that the extra membrane is recycled through endocytosis, the team filled the synaptic cleft with horseradish peroxidase, also abbreviated as HRP. HRP is an enzyme that produces a dense reaction that can be visible with an electron microscope as this black dense material. These images can be a bit confusing though, because of the fact that mitochondria also appears black in electron micrographs here shown in blue. Anyway, as you can see in figure 20, when the terminal was soaked in HRP for 30 minutes, the compound surrounded the terminal and the scientists were even able to capture a synaptic vesicle with HRP in it here shown by the green arrow. In figure 21, the terminal was stimulated for a minute and caused HRP to enter structures now known as endosomes. Here again, the scientists only saw one vesicle filled with HRP. The scientists now performed a stimulation for 2 and 15 minutes on different terminals, and you can see the results in figure 25 and 26. The pictures are not too different from the previous experiments, except from the fact that there are more endosomes and vesicles filled with HRP. Now, it gets more interesting at figure 27, where Reese and Heuser stimulated the terminal for 15 minutes, rested it for 1 hour, and washed the extracellular space with any HRP. As you can see, a large number of HRP vesicles now appear in the terminal. As they say, these vesicles presumably come from endosomes. But one thing for sure, these results illustrate and support the idea that, with presynaptic stimulation, the extra membrane added onto the terminal, as a consequence from fusion, is rapidly recycled through endocytosis and allows to subsequently produce new vesicles and maintain the vesicle population without depleting the stocks too quickly. From these two papers of Heuser and Reese, along with the precedent work made by Bernard Katz and other scientists that I've covered such as Rodolfo Linas, it solidified the idea that the quanta are vesicles that are released upon a calcium signal and that after their exocytosis, they are recycled through endocytosis. As time went by and the technology used in biology exponentially grew, many more molecular studies about the proteins involved in the life cycle of a vesicle have allowed scientists to come up with a general model of what the cycle is. The vesicle cycle begins with neurotransmitter uptake. For small molecules such as acetylcholine, the uptake is facilitated by an active transport mechanism. The vesicles have a vacuolar-type proton ATPase, more commonly referred to as a VATPase, that uses the energy generated by the hydrolysis of ATP to pump protons in the vesicle. Respective transporters for the neurotransmitter then use the energy source of the proton gradient made by the VATPase to get neurotransmitters inside through an antiport mechanism. The transporters are specific to the neurotransmitter. For example, the transporter for acetylcholine, the vesicular acetylcholine transporter, is different from the vesicular glutamate transporter, which imports glutamate, another neurotransmitter we will cover later. The filled vesicles are then moved into a reserve pool just outside the active zone. A protein named synapsin holds the reserve pool stable by cross-linking the vesicles together. Synapsins are substrates of two kinases, protein kinase A and calcium calmodulin-dependent protein kinase. Upon phosphorylation by the kinases, the synapsins are released from the vesicles and allow the vesicles to enter the next step. The filled vesicles that move near the plasma membrane, that are not held together at the reserve pool, undergo docking and priming to the membrane. Given the stability of the plasma membrane, it takes a lot of energy to fuse the vesicles. To overcome this energy, vesicles form large protein complexes with the membrane. One of the most important and recurrent classes of proteins that mediate diffusion are referred to as snare proteins and constitute the core machinery that performs fusion. Snare proteins come in two main types depending on where they are. Snares that are on the vesicles are referred to as V snares, and if they are on the target membrane for fusion, they are referred as T snares. Each synaptic vesicle has a V snare called synaptobrevin, and in this case, the target membrane is the plasma membrane, and it has two distinct T snares, syntaxin and SNAP25. At the docking stage, these snare proteins all collectively bind together to form a tight alpha-helical coil-to-coil complex. Another important protein in the system, named MUNC18, binds to the complex and allows its formation. Normally, this complex on its own can fuse the vesicle to the membrane, but a protein named complexin joins the complex and prevents that spontaneous fusion. From what we've discussed, given that calcium is the signal for vesicle release, the system must have a protein that couples the influx of calcium to the fusion process. The class of proteins that mediate this signal are part of the synaptotagments, which also join the complex. Now that the vesicle is docked and primed for fusion, it sits at the membrane waiting for calcium. Remember that voltage-gated calcium channels are mostly localized at the presynaptic terminal and that calcium is highly concentrated at the extracellular side. The leading hypothesis on how fusion works stipulates that as the action potential comes and depolarizes the terminal, the depolarization opens voltage-gated calcium channels and lets calcium enter. After entry, calcium binds to the binding sites of synaptotagmin, which dislodges the complex in such a way that complexin is removed and allows the exocytosis and lets the neurotransmitters diffuse in the cleft. As you can see, this system allows the fusion process to be very quick as calcium can enter and immediately cause the fusion of the vesicles. If the vesicles weren't already docked and primed, the delay in chemical transmission would be much higher. After fusion and transmitter release, calcium will unbind from synaptotagmin and its activity will be terminated by either directly leaving the cell through transporters or by getting buffered by specialized proteins. The calcium entry will also be removed by the repolarization of the cell by voltage-gated potassium channels. Neurons sometimes have to release high amounts of neurotransmitters, and to prevent rapid depletion of their stock, it is considerably more convenient for them to directly recycle the vesicles and the proteins associated to them at the axon terminal rather than to synthesize the vesicles again way back at the cell body. Regardless of how the vesicles are recycled, the snare complex must be disassembled after fusion by two important proteins, named NSF and SNAP. NSF is an ATPase that binds to the complex via an adapter protein SNAP and dissociates the complex with the energy released by ATP hydrolysis. One common route for vesicle recycling is clathrin-mediated endocytosis. Clathrin is a trimeric protein that binds to the membrane through adapter proteins and assembles into a cage-like structure. This cage-like structure dramatically changes the curvature of the membrane and forms the foundations for a new vesicle that is still associated with the membrane. A protein by the name of dynamin is a GTPase and uses the energy from GTP hydrolysis to pinch off the membrane by forming a ring around the lipid stalk. This subsequently frees the clathrin coat vesicle. The clathrin coat can then be removed by other proteins such as HSC70 and oxalin. From there, the free vesicle can either directly get refilled and restart the cycle, or it can join the endosome. From the endosome, a new vesicle can form and get filled with transmitters and restart the cycle from there. Another possible route for recycling is through the reversible fusion pore mechanism, which occurs after the fusion process. Given the high quantity of proteins associated with the vesicle, it is good to ask ourselves how do the vesicles manage to maintain all of these proteins in the next rounds of release if the vesicle totally fuses with the membrane. In the reversible fusion pore mechanism, when the vesicle fuses at step 4, it only fuses partially through an opening called the fusion pore. After snare dissociation, the fusion pore can then rapidly close and reform the vesicle. This process of partially opening the vesicle and then closing is also referred to as a kiss. If, after reformation, the vesicle stays at the membrane, the vesicle is said to do the kiss and stay pathway. If the vesicle goes back to the endosome or gets directly refilled, the vesicle is said to do the kiss and run pathway. Nonetheless, the clathrin-mediated vesicle recycling pathway is more commonly taught as being the default mode of recycling, so we will make the same assessment for a model and consider the clathrin mechanism as our default. With this being said, the vesicle cycle as we know it looks a bit like this. In summary, a lot of proteins are involved at each step of the vesicle cycle, and they each have their role to play to make the vesicle cycle an efficient process. Any deficiency in any of these proteins will result in hindered chemical transmission. Obviously, this is not an extensive list, and many more proteins contribute to the efficiency of this process. For example, if we consider the proteins on the vesicle, we've only scratched the surface of all the proteins that are there and contribute to the cycle. With the vesicle cycle now covered, when we look back at the neuromuscular junction, we have a pretty good picture of what's going on presynaptically. From the action potential to the release of neurotransmitters in the cleft, we've covered a good deal of what's experimentally significant to support the chemical synapse model. But the last detail for us to consider in order to have a complete picture is to discuss the properties of the postsynaptic terminal. Indeed, we know for the moment that the end plate potential that occurs as a result of acetylcholine release in the cleft is depolarizing and causes an action potential in the muscle fiber. But we still need to determine which ions flow through the nicotinic acetylcholine receptors to cause that depolarization. To determine which ions go through these postsynaptic receptors, we can perform a patch clamp experiment on them. Remember that the patch clamp experiment has 4 different configurations that all have their respective benefits. For our inquiries, the outside-out patch clamp configuration is the better suited configuration because it allows us to change the extracellular medium, and that's very helpful when dealing with ligand-gated channels. When we consider the outside-out configuration in more detail, what we are essentially doing is a voltage clamp on a small patch of membrane that contains a single channel. In other words, we are locking the membrane potential of the channel at a specific command voltage, and then we measure the current going in or out depending on the extracellular conditions we subject the channel to. Recall that we've used the patch clamp experiment to determine the properties of leak and voltage-gated channels in our conversation about the action potential. To make sure we're all on the same page, let me briefly give the key insights that we can get from the patch clamp experiment by using our previous work. When we consider the patch clamp results from the voltage-gated channels, the most important aspect from the recordings is that the individual current coming from one channel, also known as the microscopic current, is highly probabilistic due to the nature of the channels, which is something that you can review in earlier discussions of the gates behind the voltage-gated channels. The total or macroscopic current will come from the summed activity of all the channels. So even though on an individual scale some channels might open for longer or shorter than expected, the total current has defined properties as it represents the statistical average of all the channels. Another important aspect is that the current going through the channels is equal to the conductance times the driving force. This is a relation that we've established rather early in our discussion, so I suggest reviewing it if it doesn't speak to you. Now, in voltage-gated channels, the conductance is dependent on voltage and time, whereas leak channels have a constant conductance. This constant conductance has allowed us to draw a linear IV plot according to the experimental results that we've gotten from the patch clamp. To quickly recap how to read an IV plot, the x-intercept corresponds to the reversal potential of the channel that is being experimented on. The reversal potential is also known as the equilibrium potential, and it corresponds to the potential at which there is no net ionic flux. In this example for potassium, the reversal potential is at negative 88 mV, and as you can see in the recordings, no net flow of current occurs. By reading the IV plot, we can also understand that when the cell is above negative 88 mV, the current is positive, which, according to our convention, means that positive charges like potassium in this case leave the cell. This current is also said to be outward. Inversely, if the potential is below negative 88 mV, then the current will be inward and potassium ions will enter the cell. Luckily for us, the conductance in ligand-gated ionotropic channels, such as the acetylcholine receptor, is not voltage-dependent and behaves more like leak channels, which we can directly see if we plot the results from the patch clamp recordings. As a quick note, for the patch clamp experiment to work, the extracellular medium must obviously contain some acetylcholine to open the receptors. Another thing that I want to point out is that even though the ligand is bound to the channel, ionotropic channels can inactivate and prevent current from flowing. In the recordings, this doesn't make too much of a difference because it simply shows when the channels are open and let current flow or closed and don't let current flow. It doesn't really differentiate between the closed and inactivated state. Alright, so the key in determining which ions go through the channel is to consider the reversal potential of the curve. As we can see from the plot, the reversal potential is at about 0 millivolts. When we consider the individual IV curves for each ion that we know are physiologically present, you will notice that the IV curve for the receptor does not match the curve for any of these ions. Hence, the acetylcholine receptor cannot be letting only one ion flow through. It turns out that the receptor is almost equally permeable to sodium and potassium. To confirm that sodium and potassium are indeed the two main ions flowing through the channel, one can change the concentrations of each ion to see how the IV curve is impacted. For example, if the external sodium concentration is lowered, the equilibrium potential for sodium will also be lowered. As a result, the IV curve for both sodium and the acetylcholine receptor moves to the left. Inversely, if the external potassium concentration is increased, the equilibrium potential for potassium increases and the curves for potassium and the receptor shift to the right. This is typically the experiment that is used to illustrate what the receptor is permeable to, but keep in mind that the receptor is also permeable to calcium with varying degrees depending on its subunit composition. Nonetheless, this is good enough to support the idea that potassium and sodium are the main ions flowing through. With this IV curve, we can now get more insight on what happens at the ionic level for the neuromuscular junction. Remember that in this figure on the left, we are looking at a close-up of the muscle fiber. But for the moment, let's just consider one open channel. When the muscle fiber is at rest, its membrane potential is at about negative 90 millivolts, which is essentially the value for the equilibrium potential for potassium. This means that when the channel opens as a consequence of acetylcholine binding, the current flowing through the channel will be mainly made out of sodium because it has the highest driving force to enter the cell. This sodium entry will cause a net depolarization and obviously impact the membrane potential. As the membrane potential increases from the resting value, the driving force for sodium decreases and the driving force for potassium increases, which makes potassium flow out of the channel more and more. This efflux-influx dynamic of sodium and potassium inevitably come to a balance at the reversal potential where the two currents are equal to one another. If the opening of the channel is further sustained and the depolarization increases, the channel would start hyperpolarizing as the potassium current would take over. Now, in physiological conditions, this potassium hyperpolarization does not occur because the brief moment at first when sodium overtakes the net current makes so that the muscle fiber is depolarized above threshold and an action potential is fired. Furthermore, the acetylcholinesterase enzyme rapidly degrades acetylcholine, which forces the channel to close and prevents the channel from reaching very high positive potentials. To add another layer of comprehension to our neuromuscular junction model, we can consider modeling it through an equivalent circuit model. Indeed, as we did for the propagation of the action potential, we can use the equivalent circuit model to get an accurate picture of the currents involved in the postsynaptic potential. To get the proper background knowledge on the electrical circuit parallels with the neuron, you can consider going back to our previous discussions where we defined the parallels and equations that can be derived from the circuit. Nonetheless, let me make a brief review of the key ideas behind the electrical circuit model so we're all catched up to speed on what's relevant. Alright, so on the postsynaptic membrane, let's consider the nicotinic acetylcholine ligand-gated receptors and the leak channels that are embedded in the membrane. To simplify the picture, we won't consider any voltage-gated channel. Along with being filled with channels, the membrane also allows to separate charges on either side by acting as a dielectric. This charge separation essentially constitutes a form of capacitance, which can be shown by this symbol, and creates a voltage difference between the cytoplasm and the extracellular matrix. From experimental recordings, the membrane potential of the muscle fiber is at around negative 90 millivolts. Due to the embedded channels, the membrane is a leaky capacitor because the channels allow charges to flow in and out of the cell. To represent the channels, we can use the resistor symbol, which has a corresponding value of conductance. For the acetylcholine receptor, I will use the subscript EPSP to make it more generalized. Now, remember that the channels are powered by both the membrane potential and their own equilibrium potential, which is represented as a battery. Here again, the value of the battery has a subscript EPSP. From a past derivation, the current going through the resistor in series is equal to the conductance of the channel times the driving force. As for the capacitive current, remember that it equals the capacitance times the rate of change of the membrane potential with respect to time. As we've seen in the patch clamp experiments, the equilibrium potential for the ionotropic acetylcholine receptor is 0 millivolts. Now, one particularity of making a circuit for the postsynaptic cell is to consider how the ligand-gated channels operate. To model the binding of the ligand to the channel, we can consider it as a simple electric switch. When the switch is open, no ligand is bound and the circuit does not operate. And when the switch is closed, it means that the ligand is bound and the current flows. With our completed circuit, let's now put it in perspective and see how the currents evolve over time with an excitatory postsynaptic potential and see what relations we can draw from it. To do so, we will consider the plot of the current and the membrane potential as a function of time. Alright, let's first consider the resting state. At rest, when there is no depolarization and no ligand is bound to the channel such that the circuit is open, all the currents are equal to zero, including the capacitive current. Remember that the capacitive current comes from charge repulsion when there is a change in membrane potential. When acetylcholine binds to the receptor, the circuit closes and causes an inward current coming from the channels. As we've seen, most of this depolarizing current comes from sodium because of its high driving force to enter. This inward current is balanced by two outward currents. First, as the membrane potential changes, there is a very quick outward capacitive current. Secondly, the leak channels, which are mostly permeable to potassium, also provide an outward current. As a result, to satisfy the law that any current going in is equal to any current going out, we can establish in this rising phase that the current from the acetylcholine receptors is equal to the negative sum of the leak and capacitive currents. At the peak of the synaptic depolarization, the inward current from the acetylcholine receptors directly equals the outward current from the leak channels. This happens at the peak of the EPSP, where the capacitive current is equal to zero. Indeed, since the capacitive current is equal to a constant times the derivative of the membrane potential, remember that purely visually, we can tell that at a critical point, such as a local maximum of a curve, the tangent line has a slope of zero and thus the derivative equals to zero. So with the capacitive current being equal to zero, we can easily isolate the membrane potential of the peak with some algebra, which gives us this relation. With this expression, we can compute what is the peak membrane potential according to whatever values of conductance we have. Now, as the acetylcholine receptors close because of acetylcholine degradation by acetylcholine esterase, the current coming from these channels progressively decrease and the membrane potential starts to repolarize. As the current from the leak channels overtakes the current from the acetylcholine receptor, the capacitive current reacts to the hyperpolarization in the opposing manner it reacted at the beginning. Hence, you see this inward capacitive current. As a summary of what we've discussed, you can see on this packed schematic the evolution of the membrane potential, the currents, and the circuits as a function of time. With the equivalent circuit model of an EPSP now explained, we have a pretty good picture of how the neuromuscular junction operates and what experiments led to this model we use today. Now, even though the neuromuscular junction is a very appropriate model to describe how chemical synapses function, it still lacks a lot of the features that synapses in the central nervous system have, which we are now ready to consider. The first main difference is the quantity of innervations that the postsynaptic cells receive. At the neuromuscular junction, the muscle fiber is usually innervated by one motor neuron, whereas in the central nervous system, neurons can receive up to 1,000 connections. Secondly, the signal transmission in the neuromuscular junction is mediated by a system of one neurotransmitter, acetylcholine, binding to one postsynaptic receptor, the ionotropic nicotinic acetylcholine receptor. As a result of this simplicity, muscle fibers only receive excitatory inputs. On the other hand, chemical transmission in the central nervous system can be mediated by many different neurotransmitters which bind on many different receptors that can be either ionotropic or metabotropic. Due to this diversity, postsynaptic neurons can receive excitatory or inhibitory inputs. Another very important distinction between the two systems is that action potentials in the neuromuscular junction will usually produce an action potential in the muscle fiber. As we've discussed, the threshold for action potential in the muscle fiber is rather low, so the effects of acetylcholine are very strong. In the central nervous system, the presynaptic inputs are generally weak, such that it takes many excitatory inputs to generate an action potential in the postsynaptic cell. Also, given the addition of inhibitory inputs on the postsynaptic cell, it adds an additional layer of integration that we will have to consider. In all, there are 5 main differences between the two systems that we shall consider in this section. Luckily for us, the work we've made with the neuromuscular junction, such as the vesicle cycle or our understanding of ionotropic channels, is still very valuable and will apply to the central nervous system. With this being said, I want to begin our conversation on neurons in the central nervous system by first describing what metabotropic ion channels are and what purpose they serve. Thank you for watching this video. If there was anything unclear or there was a mistake somewhere in the video, make sure to let me know in the comment section. If you enjoyed this video and found it useful, you can consider leaving a like and subscribing to support the channel. On the right, you will see the informational resources that I've used to produce this video. Thank you again for watching, and I'll see you in our next discussion.